Hi, today we'll talk briefly on animal reproduction and animal development. The development starts with maturation of the adult that soon do spermatogenesis and oogenesis to produce sperm and ova. After fertilization, it is the merging of the and fusion of sperm and ova. The zygote is formed and successive cleavage, successive of divisions occur termed cleavage, which lead on to gastrulation. In gastrulation, the embryo tries to be a double-layered and then triple-layered embryo. After gastrulation, neurulation starts, which is a process in which the formation of neural tube. This is followed by organogenesis, then cytodifferentiation, then individual life that grow to reach maturation. Developmental time for a cell to be a specific cell type is accompanied by restriction of other fates, possible other fates for the cells, then further restriction of possible fates, then determination of a specific cell type, then fully functional cell is formed. This restriction and determination is controlled molecularly by the expression of genes associated with these fates that have been restricted, then expression of a further expression of genes, other genes associated with these fates, then expression of many genes associated with the final fate, and then the fully functional cell will be differentiated. So let's look closely for these embryos. So we have 6 embryos. What do you think? Which embryo is the human embryo? Do you think it's a fair A, B, C, D, E, F? Actually, the human embryo is C. So basically, you can see that a lot of similarities between different embryos. And of course, E is the embryo, is the hypothetical embryo. It's not a real embryo. As you said, the process of development starts with gametogenesis. So gametogenesis either oogenesis or spermatogenesis. In oogenesis, it starts by the primary sex cells, which is the oogonia. This oogonia, in the process of mitosis, increase in number. This followed by first meiotic division to produce secondary oocyte, and then the second meiotic division, and they had to be arrested at metaphase of meiosis 2 until ovulation. Soon after that, one oocyte is developed in the meiosis division 2, and the two polar bodies is formed, one from meiosis 2 and the two from meiosis 1. For spermatogenesis starts with spermatogonia, and this spermatogonia divide mitotically to give a spermatogonia type A, then type B. Then it will be commitment to give primary spermatocyte in prophase meiosis 1 and still diploid. The meiotic division resulted in a secondary spermatocyte, which is haploid, then a spermatid, then a process very specific for male spermatogenesis, which is called spermiogenesis, in which the spermatids are transformed and metamorphosed to be a spermatozoon, and this is the active mobile sperm. Once the sperm and ova is formed, the embryogenesis starts after the process of fertilization. So the gametogenesis is a process by which the diploid germ cells give rise to haploid sperms via spermatogenesis or ova via oogenesis. And it is divided into 3 stages. Multiplication phase, in which the germinal epithelium cells divide mitotically to give rise to spermatogonia or oogonia. Growth phase, which each of these tiny cells produced by multiplication grow to large sites called primary spermatocyte or primary oocyte. Maturation phase, during which chromosome number is reduced to its half original number, reduction division of meiosis. So the ovum have 3 functions to perform: supply a nucleus with half chromosomal complement for future embryo, supply almost all the cytoplasm, supply food reserve to enable the embryo to develop to the stage where it can begin to feed upon exogenous material. Therefore, the animal ovum is a cell of gigantic size. The sperm has to find its ovum. To accomplish this, the sperm is equipped with the organ of locomotion and with high rate of metabolism so that it can generate energy to move rapidly over a great distance, over relatively great distance. Once in contact with the egg, it must provide a lytic substance to dissolve the ovum outer membrane, delivering a nucleus containing a haploid set of chromosomes, one half of the complement of the future embryo. The sperm is morphologically divided into four parts. Head, which contains acrosome and large nucleus, and this acrosome aids in the penetration and activation of egg. Neck, it is the location of two centrioles, the proximal and distal centrioles. The distal centriole gives attachment to the tail of the axial filament of the sperm tail, while the proximal centriole have a key role in the division of the fertilized egg. Then a middle piece formed entirely of mitochondria, mainly for metabolic and energetic aspects of sperm function. Then the tail, which actually the axial filaments, forms the main element of the sperm tail. It provides motility for the sperm through whipping the tail. You can see here a figure representing sperm and ova, and you can see the regions of the sperm, while the ova have mostly cytoplasm and surrounded by a membrane called the vitelline membrane, and have follicular cells for nourishment around the ova. This is a nice photo showing the process of fertilization and how sperm try to penetrate the egg. So as a coda, the elements that unite are single cells, each on the point of the death. That means if the sperm will not unite with the ova or the ova will not unite with sperm, both of them will die. But the reunion, rejuvenated individual, is formed, which constitutes a link in the eternal process of life. Here you can see it's a real image of how sperm trying to penetrate the egg vitelline envelope to do fertilization. This as well, an E-image, electron microscope image, showing the different, actually it's a section in the sperm showing the different organization and different region of sperm. As well, this is the same, this EM micrograph or figure showing the internal structure of the ova. You can see that a lot of it's a cytoplasm, you have a cortical cytoplasm, and you have mitochondria is around located internally, centrally, mainly centrally. This you can see that the first one is the normal sperm, but you can see a lot of abnormality of sperm can arise through the process of spermatogenesis. So the human male ejaculates around 200 to 300 million sperm during coitus. 60% must have a normal shape. 40% must show vigorous motility. And this is the minimum requirement for the sperm for the total sperm to be able to fertilize. You can see here in oogenesis in mammals that most of the multiplication and maturation occur inside the ovary. Starting with the primary follicle, then this primary oocyte develop into developing follicle, mature follicle with a secondary oocyte, and this is ruptured, releasing the secondary oocyte, and it will wait until the sperm comes and fertilize. Once there is, the oocyte is released, a corpus luteum is formed. This is a nice comparison between spermatogenesis and oogenesis. You can see the location, then the number of gametes produced, fewer of course in case of the female, and then the gametes per germ cells, 4 in male, 1 in female, and the beginning of the process. In spermatogenesis, it begins at puberty, while in oogenesis begins during fetal development, while it is inside the womb. The timing, it's continuous for male anytime, but the female is controlled. There is a once a month, actually a menstrual cycle. The endo process, the fertility is lifelong but reduced. Fertility in female, fertility stops at menopause. The timing, any time for the spermatogenesis, while for the female oogenesis is monthly cycle. The meiotic division uninterrupted, but in oogenesis the meiotic division is arrested until the sperm comes in contact with the ovum. The germline epithelium is involved in gamete production, while in oogenesis is not involved in gamete production. So fertilization. So now we have sperm and ova. So fertilization is the process whereby two sex cells, gametes, fuse together to create a new individual with genetic potential derived from both parents. Fertilization accomplishes two separate events: to transmit genes from parents to offspring and to initiate reaction in the egg cytoplasm that permit the development to proceed. Generally, fertilization consists of four major events: sperm and egg contact and recognition, regulation of sperm entry into the egg, only one sperm can fertilize egg, fusion of genetic material of sperm and egg, activation of egg metabolism to start development. So you can see here animated image. The first figure is a scanning electron microscope seeing a lot of sperms trying to penetrate the egg, while the second image showing sperm approaches, penetrates the egg, fusion of the genetic material, start of a successive, after that successive cell divisions, 2, 4, a lot of cells, and then proceed in development. So again, so we reached cleavage. So cleavage is fertilization is followed by cleavage, which actually is a series of mitotic division. Actually, the enormous volume of zygote cytoplasm, because now we have a zygote which is full of a lot of cytoplasm. During cleavage, this enormous volume of cytoplasm is divided into smaller cells called blastomeres. The zygote genome doesn't function in early cleavage embryos. First, the zygote is divided into half, then quarter, then eighths, and so forth. The division is accomplished by abolishing the growth period between cell division, i.e. no G1 and G2 phase of cell cycle. So the cleavage of the nuclei occur at a rapid rate, never seen again. So you can see here how the cell cycle have changed from the normally G1S, G2M into S and M. There is no G1 and G2. So during cleavage, and this is a cell cycle of blastomeres, as I said, there is no G1 and G2. So it's a very fast cleavage, resulting by the end that the cells, the blastomeres get smaller and smaller and smaller because there is no growth phase. So actually the cyclin, the group of protein synthesis, allows the progression to M, while cyclin degradation allows cells to pass into S phase. Cells that are differentiating are usually taken out of the cell cycle and are in an extended G1 phase called G0 or G0. Mitosis is controlled by the cytoskeleton, so we'll deal with the cytoskeletal mechanism briefly of mitosis. Cleavage is a result of two coordinated processes, karyokinesis and cytokinesis. Karyokinesis is the mitotic division of the nucleus. The mechanical agent in this process is the mitotic spindle, which actually is a tubulin protein. While the cytokinesis, it is a division of the cell. The mechanical agent is a contractile ring, as you can see from the figure. It consists of actin filaments. It's another set of protein. The mitotic spindle and the contractile ring must be perpendicular to each other. The contractile ring creates a cleavage furrow which bisects the plane of the mitosis, thereby creating two genetically equivalent blastomeres. And you can see here the process, it's a comparison between karyokinase and cytokinesis by the mechanical agent, the major protein, tubulin and actin, the location, and the major drugs used to stop this process. Colchicine is used to stop karyokinesis, and cytocalcine B is used to stop cytokinesis. And these drugs are used as well for treatment of cancer. The patterns of embryonic cleavage. So different organisms undergo cleavage in different ways. The pattern of embryonic cleavage particular to a species is determined by two major parameters: the amount and distribution of yolk, actually the yolk protein within cytoplasm. Second, the factors in egg cytoplasm that influence the angle of mitotic spindle and the timing of its formation. So the amount and distribution of yolk determines where cleavage can occur and the relative size of the blastomere. When one pole of the egg is relatively yolk-free, the cell division occurs there at a faster rate than at the opposite pole. Normally we say the yolk-rich pole of the zygote is a vegetal pole, while the yolk-less pole is the animal pole. The zygote nucleus is frequently displaced toward the animal pole, which is obviously because it's have less dense and less yolk in there. In general, yolk inhibits cleavage. Continuing the patterns of embryonic cleavage, eggs of sea urchin, mammals, and snails, they have sparse and equally spaced yolk. This type of egg is called isoleucecil egg. And this type of egg undergo a type of cleavage called hollow, means complete, holoplastic, so complete cleavage of the blastomeres. So the cleavage furrow extends through the entire egg. Embryo must have other ways to obtain food, because food will not, since it is sparse and very few, it will be enough until the final stage after hatching. So the yolk for these kind of embryos is enough only until reaching a larval stage, or the embryo must be attached by placenta to the mother. In the absence of large concentration of yolk, there are 4 types of holoplastic cleavage can be observed: radial holoplastic, spiral holoplastic cleavage, bilateral holoplastic cleavage, and rotational holoplastic cleavage. In case of moderate vegetal yolk, moderate amount of yolk, the type of egg is called mesoleucecil egg, and as well it undergo a holoplastic cleavage. That's mean complete cleavage. That's mean a division path through whole egg. Eggs of insects, fish, reptiles, and birds, most are full up with yolk, made up of yolk. So they undergo another type of cleavage called meroplastic cleavage, in which a portion of the cytoplasm is cleaved, not the whole egg. Cleavage furrow doesn't penetrate into the yolky portion of the cytoplasm. Yolk is sufficient in this case to nourish this animal until it hatches. So the hatching of the young will resemble the adult, but it needs time to grow and mature. Eggs of insects have yolk in the center. This is another type of egg called centroleucecil, in which the yolk is concentrated in the center. So cleavage occurs only at the rim of the cytoplasm. It doesn't penetrate into the inside of the egg. So this type of cleavage is called superficial cleavage. The last type of egg is egg of birds. Fishes have only small area of egg that is free of yolk, and most of the ovum is yolk. So this type of egg is called the teloleucecil egg. Cleavage occurs only on a small disc of the cytoplasm, and this type of cleavage is called discoidal cleavage. You can see here the types of holoplastic cleavage. In case of the isoleucecil egg, you can see the radial cleavage, then spiral cleavage as in annelids, mollusks, and flatworms, and then the bilateral cleavage, and then the rotational cleavage. So you can see that there are different patterns of cleavage for each type of an organism, but all of them share they share two important features. They are isoleccal, that means the yolk is sparse, few, and evenly distributed. And second, that the egg is divided whole, not a part of the egg is undergo cleavage. The whole egg is cleaved. The second type of egg is mesolithal cleavage, which actually contain a moderate amount of cleavage, and it is located in the lower hemisphere, which we call it vegetal hemisphere. So you can see that the furrows, especially the transverse furrow, is shifted a little bit upward because there is a lot of yolk in the bottom half. In a merocloblastic cleavage, that's for a telerecessal egg, you can see that only cleavage occur on top of the egg. So you have a bilateral cleavage or a discoidal cleavage. So the whole egg, that's why it's not holoplastic, because not the whole egg is divided. The last type is the centrorecessal egg, in which the yolk is concentrated in the center, and you can see that the blastomere that arise from the cleavage in the last figure are all concentrated at the periphery. That means the cleavage is superficial around the yolk. You can see here how the embryo is starting first cleavage with a cleavage furrow, then the second cleavage which is perpendicular to the first one, and then the third cleavage which is horizontal. This is scanning images, scanning electron microscope. Gastrulation. Cleavage ends by formation of a stage called the blastula stage. The definition of a blastula stage, it's a numerous cells that position of which were established during cleavage. Gastrulation is a process by which cells of blastula are dramatically rearranged. During gastrulation, these cells are given new position and new neighbors, and the multi-layered body plan of the organism is established to form a gastrular stage. So cells that will form endodermal and mesodermal organ are inside the embryo, while cells that will form ectodermal organs are spread over its surface, outside surface. So that's why we said during the process of gastrulation, cells are given new position and new neighbors. Endodermal, mesodermal inside the embryo, ectodermal cells which will give ectodermal organ are spread outside the embryo. So basically, thus three germ layers are produced during gastrulation: outer ectoderm, inner endoderm, and the interstitial mesoderm. From these three germ layers, you can see that lung, thyroid cells, and digestive cells arise from the endoderm. Muscles, tubule cells, red blood cells, and arise from the mesoderm, while the ectoderm will give skin, neurons, and pigments. Of course, these are some of the derivatives of the germ layers. Here you can see an animated image showing the successive stage of development of a salamander larva. Salamander is an amphibian organism. So you can see it starts with the cleavage. Now it's in the process of neurulation, formation of a neural tube, and try to formation the part that will form the brain. Again, you can see cleavage 1, 2, 3, a lot of successive cleavage. Blastomere arise, smaller blastomeres at the top, since the yolk is a moderate amount at the bottom. So that's why blastomeres are smaller on the top than the bottom. Then again, the process of this neurulation, you can see again when cleavage. At the time there is a lot of cells covering the embryo, that's a blastomere, a blastula stage. Blastula stage then start gastrulation, and this is the rearrangement of cells to internalize the endoderm and the mesoderm, and then followed by the neural tube formation, as we said, and then embryo will keep going for organogenesis. So we'll talk briefly about the development of the human embryo. So the cycle of development starts with two mature adults giving sperm and ova, forming fertilization, which is smallest interval in the life of the human embryo. Then this fertilization results to a zygote that undergo a cleavage until forming two cell stage, four cell stage, morula stage. It's a mass of cells with no cavity. Then the blastocyst consists of two types of cell. Then starts the process of gastrulation, formation of gastrula, which end with the beginning of the organogenesis, and at week 5, 6, and then week 5, and then 8, and then before birth. The process from that constitute cleavage, gastrulation, and organogenesis are all called embryogenesis. And here you can see the sperm and the ova scanning image showing the sperm try to penetrate the ova. The process of penetration of a sperm to the ova is divided into three phases. Phase 1, actually the sperm undergo acrosome reaction and penetrate the corona radiata, the cells around the ova, while phase 2, the sperm penetrate zona polyosida, the region around the ova, while phase 3, the sperm and oocyte plasma membrane fuse, and now the nucleus is inside. The nucleus of the sperm is inside to fuse with the ova. So in cleavage, you can see that it begins 12 hours post-fertilization. The zygote divides into two cells by mitosis. The 46 chromosomes in zygote equal 46 chromosomes in both daughter cells because it's mitotic division. The two cell into four cell stage in 36 hours max. The four cell into 8 cells in 72 hours max. And then we call it the 16 cell stage is called morula stage. Here you can see 72 hours post-fertilization, the embryo enters the uterus to get implanted inside the wall of the uterus. So you can see that the sperm is fertilized in the upper third of the oviduct. So the sperm migrate from the vagina to the uterus to the oviduct until reaching the first third of the oviduct to undergo fertilization. Once fertilized, the zygote start to move, undergo cleavage until reaching the uterus. It had to reach the stage of blastocyst before entering the uterus because the blastocyst stage will help the embryo to implant itself inside the wall of the uterus of the female. Here you can see briefly through the week 3 and week 4 how the development of the embryo and the increase in size during development and the implantation inside the wall of the inside the wall of the uterus to form the placenta. Now the embryo at week 4 very obviously attached to the wall of the uterus through the umbilical cord, which contain the umbilical vein and umbilical arteries. And you can see the yolk sac, which uses to feed the embryo. And now the embryo is covered by the amniotic, the amnion, which encloses the embryo in amniotic cavity. Embryo fetus. So the embryonic period is a week from week 1 to 8. Fetal period begins on week 9 and goes until birth at week 38. Embryonic period is characterized by development of organs, while fetal period is characterized by growth of those organs. Thank you.